Go.
I remember.
Oh, what's that?
So you said last time I can't inject myself with chlorophyll to make my own energy.
No, it doesn't work that way.
So where do I get my energy from?
Okay, you eat, and the food has energy that your body breaks down.
But where do the plants get the energy?
Oh, we talked about this.
Oh, we did. Okay,
so,
that's right.
Energy from the sun is converted into chemical energy in the form of glucose. You eat plant products. You eat plant products, and then your body breaks down all those bonds in glucose, releasing energy for your body to use to do work. So ultimately, the sun is where you get all your energy from. Ultimately.
But you want to know somewhere else I get my energy from?
Where?
From exercising.
No. You may feel energized after you exercise, but exercising is burning energy. So you have to use glucose, and you have to use cellular respiration to have energy to exercise.
But Mr. Young runs like 16 miles a day, and he's got tons of energy.
Yeah, he also eats 10,000 calories a day. And those calories come from food, and food gets broken down, so therefore he has energy to power his 200-mile runs.
So if he didn't eat, he wouldn't run?
No. This may come as a surprise to you, but hopefully it's not. Organisms get the energy they need from food. Food molecules contain chemical energy, and it's released when bonds are broken between molecules. A calorie is the amount of energy needed to raise the temperature of 1 gram of water by 1 degree Celsius. We also use calorie, which is heat energy, to represent a food calorie. So 1,000 calories equals 1 food calorie. A gram of glucose below this table shows you a comparison between carbohydrates, triglycerides, glucose, proteins, and fat, what the difference in calories, food calories, is between them. You can see that a gram of fat has the most calories, and a gram of glucose and carbohydrates are just about tied, because carbohydrates are glucose, glucose are carbohydrates. Food molecules gradually, and they use the energy stored in the bonds to produce ATP, and ATP is going to do all the work in the cell, whether it's mechanical or chemical or transport work. Cell respiration is the process that will release energy from food. In cellular respiration, you're going to need oxygen in order to do this. The equation is 6 oxygen plus glucose, C6H12O6, will break apart to form CO2, water, and energy. So we are breaking apart our glucose. We are doing that in the presence of oxygen to form carbon dioxide and water, which are your waste products, but most importantly, it also is going to form energy. The cell has to release this energy very gradually. Otherwise, if it just released all the energy at once, you'd go up in a big ball of flames. Think of, it's equivalent to taking a match to a container of gasoline. That would be the equivalent of what would happen if this process weren't done in small steps. And that would be bad for the organism, obviously. There are 3 main steps of cell respiration. This diagram does a great job of showing us the difference. Glycolysis is the first step, and it produces just a small amount of energy, and this happens in the cytoplasm of the cell, so outside of the mitochondria. The Krebs cycle, also known as the citric acid cycle, I like Krebs cycle better because it's easier to say, is where you get a little bit more energy, and this does happen within the mitochondria. You're converting pyruvic acid, which was formed in glycolysis, into other molecules. And then finally, the electron transport chain. This produces most of the energy in cell respiration. It uses oxygen, which is a powerful electron acceptor, and you can use these electrons to do work within the cell and produce energy. The Krebs cycle and electron transport chain are anaerobic. All right, let me try that again. The Krebs cycle and electron transport chain are considered aerobic, which means they need oxygen to happen. Both of these will take place inside the mitochondria. Glycolysis, however, is anaerobic, so it doesn't need oxygen to occur. And glycolysis doesn't happen in the mitochondria, it takes place in the cytoplasm. Okay, there is a relationship between photosynthesis and cell respiration. The two relationships are, first, that photosynthesis removes carbon dioxide from the atmosphere. So photosynthesis needs carbon dioxide to start, and cell respiration creates carbon dioxide as a product, so it puts it back in the atmosphere. Second, photosynthesis will release oxygen into the atmosphere, and cell respiration will use photosynthesis product as a reactant in their reaction to create energy from food. So this diagram does a great job of showing us that photosynthesis, we use CO2 and water to produce glucose and oxygen, and these two products of photosynthesis are the reactants for cellular respiration. So to start cellular respiration, we need glucose and oxygen, and the products that we create are water and carbon dioxide. So photosynthesis will deposit energy, and cell respiration withdraws energy. They are opposite processes of each other. All right, so now we're going to get into the nitty-gritty of cellular respiration. This diagram does a really good job of showing us what reactants we start with, what products we form, where those products are going to travel in the cell, and where each process takes place within a cell, and whether we create energy from it. So often we'll refer to this diagram as we go through step by step. As we said before, glycolysis is the first stage of cellular respiration. As we go through this, the most important things to know are location, location, location. Where is this process happening? What does it need to start? And what is it producing as a result of this process? So, your main ideas here. Glycolysis is happening in the cytoplasm of cells. Remember, it's anaerobic, so it doesn't need oxygen. The reactants it needs to start are glucose. We're going to need that high-energy sugar produced from photosynthesis. We also need 2 ATP. We're going to have to power this process. It's going to cost us 2 ATP, which once used and the energy is released from ATP, we form ADP. We also need 2 NAD pluses. NAD plus is an electron carrier. We are going to take the electrons and we're going to carry them off to a later process in the cell. Now what we form, and a good way to know what you're starting with and what you're forming, is by looking at the arrows. What you start with is at the end of the arrow, and the arrow points to what you form in the process. So if you're trying to interpret a diagram, that's the way to go. So as a result of glycolysis, we produce 2 pyruvic acid from 1 glucose. We produce 4 ATP. Notice that since we use 2 ATP, we're really only gaining 2 extra ATP because there was a cost. And we're also producing 2 high-energy electron carriers, 2 NADHs, which will be used later on. Keep in mind, whatever you're producing in this stage is going to be a reactant for later stages. So pyruvic acid is a reactant in the Krebs cycle, which is the second step. Okay, let's focus on how we produce these high-energy electron carriers and then what we're going to do with them later on. So nicotinamide adenine dinucleotide, NAD+, is your high-energy electron acceptor, and it's going to take a pair of high-energy electrons along with a hydrogen, and it's going to carry those to the electron transport chain, where it's going to drop off those high-energy electrons, and they can be used to make more ATP. So remember, your high-energy electron carriers are simply a shuttle for electrons. Glycolysis will produce ATP fairly quickly, which is good because if you are running from, say, a honey badger or any other predator, you're going to need energy really fast. So glycolysis is going to provide that energy that you need very quickly. It doesn't require oxygen, so it can supply energy to cells when oxygen is unavailable. The second step is the Krebs cycle. We take pyruvic acid and we break it down into carbon dioxide. So here's where one of our products of cell respiration is produced. And it's also known as a citric acid cycle, but like I said, Krebs cycle is way less syllables, so I tend to like that one better. Okay, before the Krebs cycle, the pyruvic acid that is created during glycolysis has to enter the matrix of the mitochondria. The matrix is kind of like the cytoplasm of the cell, except for it's the cytoplasm of the mitochondria. NADP, or NAD plus, will accept 2 high energy electrons and a hydrogen, so it forms NADH. Carbon dioxide is also produced. It is one of the products, and that is a waste product that we will exhale. Any animal will exhale that waste product. And acetyl-CoA is also formed. Acetyl-CoA is then used in the Krebs cycle. So notice that all of this that we just discussed is happening before we even start the Krebs cycle. So as that pyruvic acid comes into the matrix, it gets converted into acetyl-CoA. We're going to pick up some high energy electrons, and we're producing an amount of CO2. Acetyl-CoA, and CoA stands for coenzyme A. Again, too many syllables, so we abbreviate. That is the starting product of the Krebs cycle. Okay, here's what you need to know. This is happening in the matrix of the mitochondria. So if we look back at our diagram, we are in the mitochondria now, so it must be an aerobic process. We need acetyl-CoA to start, which we got from pyruvic acid. We need 2 NAD pluses. Now FADH is new, or FAD is new to us. That is another type of electron carrier. Does the same thing, picks up high energy electrons and a hydrogen, carries them over to where they're needed later on. We also need ADP and inorganic phosphate. What we produce are 2 CO2s, so here's our CO2s being produced, 1 ATP, so that ADP and the inorganic phosphate, a bond is formed, so we store energy. That ATP can be used to do work later on. 2 NADHs, so I form an NADH here, pick up a couple electrons, and an NADH here, and 1 FADH2. Now these products are not going to travel out of the cell. They're going to actually travel to the electron transport chain, where we can use those electrons to do work. Keep in mind, 1 turn of the Krebs cycle is for 1 acetyl-CoA, but glycolysis and glucose will produce 2 pyruvic acid, which means we're actually going to produce 2 acetyl-CoAs for every glucose molecule. So the Krebs cycle is going to turn twice. So all of our products are actually times 2. The last step is the electron transport chain and ATP synthesis. The electron transport chain uses all of those high energy electrons that your electron carriers pick up from glycolysis and the Krebs cycle, and they're going to convert ADP into ATP. So the main stuff you need to know, this is happening in a membrane. Anytime you have electron transport, you have to have that separation, that membrane, so that you can pump hydrogen ions across the membrane and create a concentration gradient. It starts with NADH, high energy electron carriers, FADH2, another high energy electron carrier, ADP, and inorganic phosphate. What we produce is water, which is a waste product of cell respiration, and ATP, which we can use to do work. Notice that we have a ton of hydrogen ions on one side of the membrane and not very many on the other. So this takes energy to pump those hydrogen ions against their concentration gradient. That energy comes from all those electrons that we collected throughout the other processes. Okay, so looking at more detail, NADH will drop off its high energy electrons and its hydrogen ion, forming NAD+. That goes back over to glycolysis. It goes back over to the Krebs cycle, where it's going to pick up more electrons to replenish that. FADH2 also drops off its hydrogen ions. It also drops off its electrons, and it turns back into FAD, which is going to travel back to the Krebs cycle, where it's going to pick up more electrons. So once we have those guys dropped off, they enter the electron transport chain. Again, it's a shuttle system, where as the electrons move through these proteins, they are doing work. They are pumping hydrogen ions to one side of the membrane, so that those hydrogen ions later on can do work. I think I already said that. I'm being redundant. But again, we want those hydrogen ions to come onto one side of the membrane, so that we can build up the concentration of them. Okay, the reason we're building up these hydrogen ions is because they are going to travel over to ATP synthase. This is the same enzyme from photosynthesis, where you send one hydrogen ion through the ATP synthase and it turns the ATP synthase. And every time that ATP synthase turns, it's going to take ADP and it's going to take phosphate and it's going to put them together to form ATP. We can send that ATP anywhere in the cell to do work. So this is our big energy generator. By the numbers, glycolysis produces 2 ATP, net 2 ATP. Krebs cycle produces 2 ATP, but it's chemiosmosis, which is ATP synthase in the electron transport chain that produces 32 to 34 ATP. This is your moneymaker. This is the guy that produces all the energy for the cell. And in total, you're going to make about 36 to 38 ATP, give or take. It's a cell. It's a living thing. It's not an exact science. And it represents about 36% of the total energy of glucose. So you're actually losing 64% of the available energy as heat.
So, there are 2 muffins. Shush! I'm trying to tell a joke! Okay, wait.
There's your blooper.
There are 2 muffins in the oven. Take 2. Mrs. Follett. Once upon a time, there were 2 muffins sitting in an oven. One muffin turns to the other muffin and says, "Oh man, it's hot in here." And the other muffin goes, "I got